So maybe, Alex, you can start with, your thoughts about these 2 days and what you think were the best key takeaways here.
Thank you, Sarine. So I think we really had a superbly exciting event. We saw a lot of enthusiasm, especially in the last panel, starting with Rommie's enthusiasm that ignited everyone. But I would say enthusiasm on the part of speakers and on the part of listeners. The workshop has been oversubscribed. I think there are a lot of tremendously interesting accomplishments and no smaller number of ideas and directions for future research. So I think, if anything, this workshop had shown how significant our field is and how bright it is, and especially for people who are thinking about what to do and what profession to choose. I think it has been tremendously educational and stimulating. So that's one big takeaway that I would like to share.
Yeah, I want to join to that discussion. Yeah, I think this is really great workshop. It's really a good amount of the presentation we actually look at, and I think it's a great discussion about the integration of computational methods and experimental validation of those. I think this is really important part. There was a lot of talks which actually show that the utility of AI and machine learning, and that was a part of the goal. You remember when yesterday in the morning we tried to open the sessions and mention the goals. I think we achieved them, right? So it's really really exciting to see biotech involved in the publication and show the data, as well as the academia to develop of that approach. Plus new trends and modalities, so integration of multi-omics data from recursion. It was a great talk. All these different slices of the data, right, which they incorporate and utilize to build the machine learning. I think this is really excited to see. Of course, binders predictions from the proteins or diffusion models, really impressive work, which David showed to us as well. Really excited and great panel discussion, Rommie. It was really great conversation you facilitate today. Yeah, it was awesome.
Thanks, Alexey. Shyam?
Yeah, I think I agree with everything that's been said earlier. Fantastic, workshop, great presentations. I learned a lot personally, given my old school training of actually wet lab chemistry. So I had some generic points which I thought were very pertinent to this particular workshop. One of the points, again, Rommie's talk about biologics and design of vaccine antigens was pretty fascinating. I think now taking that particular concept model and expressing them in the right cell lines and expressing them at the yields, I think those are the next questions. The happy medium of expressing these particular molecules would be an important aspect, I think. I thought repurposing and identification of new targets and repositioning of these drugs, finding that information using AI machine learning tools was pretty fascinating. Again, that is something that I assume would be used more broadly because of the prior information that is already there, and therefore repositioning of some of the older drugs would be a fascinating area, I think. As a developer myself, I've always looked at process is the product kind of a thing, and therefore, bringing these machine learning tools and AI models, how do you develop a product really becomes an important aspect. Can it scale up once it goes from the modeling aspect to a lab to a GMP manufacturing site? I think those are important areas to look into. How much is that translation? Are these molecules able to scale as is being said, or are there issues in scaling up and manufacturability of some of these molecules? That is something to watch out for, I think. And I'm sure people are already doing this and are trying to bridge that gap. The other point is, I thought seems like the AI machine learning aspect of it, or deep learning aspect, or computational aspect, is really outpacing the clinical development. And I worry that there might be a regulatory learning, significant regulatory learning that needs to happen, which I think is probably, there is a lot of learning that's happening upfront, but downstream that gap, I think with more of these models coming up, that gap is going to increase, I think. And I think trying to bridge that gap or narrow that gap would be an important aspect, I think. And then again, prediction of toxicity, potency, PKPD, can undruggable targets be drugged, off-target effects, and understanding the critical quality attributes of these various molecules that are being modeled would be an important facet, I think. I think those would be important to really monitor. So I think I look at it completely from a product development, wet lab chemistry, or a biology point of view. And I think bridging that gap is going to be an important aspect. One of the other points I would just quickly want to make is that this is not going to just be an issue for vaccines and therapeutics, but even from a diagnostics point of view, a lot of variables and other devices do use algorithms and machine learning tools, right? And therefore, the regulatory path forward for some of these new technologies is going to be really critical, I think, to observe. I think that was my only, those were my high-level points, but really fascinating meeting. I learned a lot. I took a lot out of this meeting. And Sarine, really thank you to you and Katz for really organizing this. This was fantastic.
Thank you so much, Shyam. That was awesome to hear, like, all your learnings and you being part of this. Rommie, I'll give you some time to also say a word or two, and then we can wrap up.
Oh, sure. Yeah, I mean, I enjoyed sort of just seeing the breadth of topics, in this space. I think to me, it's just astonishing how much is going on, and it's just the whole pipeline at the moment. So it was really, I think, beneficial to, hear from folks. I'm really excited about a lot of what we heard, especially the open science initiatives. I think that's huge. We have to really get such a better grip on the data, especially. And, there's a lot of movement, and sort of things happening there. So I'm particularly, I'm happy to hear it from all these different angles, and I had a lot of fun. So thanks for also organizing this, and for the participants and speakers.
Awesome. Thank you, Rommie. So I will echo that. This was excellent. It was 2 days of learning for me as well. So wonderful talks. Thanks for all of our speakers. And also thank you all. It was wonderful working with you and our attendees as well. So I will wrap up with one more slide here. So if you're still here, thank you for hanging until the last stretch. We appreciate you so much. We would love to hear your feedback, okay? Did you enjoy this event? How we could have done this better for you? So this is a QR code here. I think the burden on this is like 2-3 minutes. Shouldn't be too long. So if you could just take this feedback survey and let us know, that would be fantastic. We'll also email you the links here. So if you can't catch it right now, you could do it later. This workshop is being recorded, so you will all receive the links to view this on demand whenever you would like. And then one more final word. I lead the Assay Guidance Manual program. This program was helped organize this event. This is multiple way you can connect with us. We do multiple other workshops as well on different topics on HT assembly discovery. So connect with us and you can learn more about our program. All right, everyone. I'm going to close it up and adjourn. Again, thank you so much, everyone, speakers, organizers, everyone. Have a good one.
Thank you, everyone. All right, bye.